All right, good morning, everybody. We're back for the final Marine Science in the Morning seminar of 2023. Today we're joined by Dean Janiak, a researcher here at the Marine Station. He's been with the Smithsonian for 14 years and started his career here, at the Smithsonian Environmental Research Center in Maryland, working in the Benthic Ecology Lab, and then in 2014 moved down to Florida to work at the Smithsonian Marine Station. Prior to that, Dean got his bachelor's in biology at Humboldt State University in California, and then did a Master of Science in Marine Science at the University of Connecticut. Today he's going to give you a brief overview of the MarineGEO program, and include some of the work he's been doing in a particular habitat that most of you might not be aware of here in the Indian River Lagoon. So, Dean, welcome.
Thank you. Let's see. Okay. Is that good?
Yep.
All right. Thanks everybody for coming. As Branson mentioned, I've been with Smithsonian now for about 14 years. I've been doing science for a lot longer than that. I remember when I first started to get into science, we never really talked about human impacts on the ocean. And it wasn't that it wasn't well understood. It's just that I don't think a lot of people knew what was going on a large scale. And I can still remember growing up in the 80s, and you would hear these phrases like the solution to pollution is dilution. And so people still thought that we weren't having that large of an impact on the ocean. And so moving well into the future, I started to get involved in how the ocean is changing and the mechanisms or the causes of that change. And it turns out that a lot of that change was from humans. And so I wanted to start with this picture, and this is from a project that I worked on last year. The idea of the project was that we were going to sample 3 local beaches, and the idea was that we picked up trash at these beaches. We counted the number of things that we found. We quantified what they were made of, if anything was living on them. And this was done at around 350 sites around the world. So it's a pretty big data set. But for me, locally, I never would have thought about how much trash that I could pick up in 30 minutes on the beaches near where our lab is. And it was pretty remarkable. And it really kind of, it was just a really interesting experience because there's just so much trash there. And a lot of this trash wasn't from Florida. A lot of this trash wasn't even from the US. I mean, this trash is coming in from ocean currents from around the world. And I wanted to highlight this for 2 reasons. The first reason is that it's a real world example that I think that everybody experiences every day. And I think that's really important because, we sort of need those experiences so that, we can maybe see this a little bit more often and try to take care of our planet a little bit better. But I also wanted to highlight this because, at least from my point of view, being a marine scientist, it's becoming increasingly difficult to study basic or fundamental ideas on how the ocean works without including something about human impacts, which is kind of a shame, but that's just the way that things are. So this brings me to a relatively new program that the Smithsonian started several years ago, and this is a program that I work for, and it's called MarineGEO. Our catchphrase, which describes the program really well, is discovering how coastal marine ecosystems work, ... to keep them working. And so this phrase has 2, it defines the program really well, and it's got 2 different ideas that make a lot of sense. The first one is discovering how coastal marine ecosystems work. And so that, to me, means that we still don't fully understand how the ocean works. And that's good for us because it keeps us excited every day to come in and figure those things out. But the other part of this phrase is that we want to keep them working, and this has got the human impact side to it. How do we keep them working with rapid global change? And that's the challenge. And so we need to figure out how they're working, but we also need to figure out how to keep them working efficiently, excuse me. And so I'm not going to spend a whole lot of time on everything that MarineGEO does. We have a nice revamped website, and the website is at the bottom on the right there, marinegeo.si.edu, and you can check it out if you're interested. But I will say that the program is, we're really interested in biodiversity, and especially coastal biodiversity. And I'll talk about what biodiversity is on the next slide. But basically, it's really easy these days to study physical changes in the ocean. It's easy to study changes in temperature or salinity. We have instruments that do that, and so it's easy to do that. But how those changes affect the plants, the animals, and the algae in the ocean, well, that's a lot harder to do, and that's what MarineGEO strives to do. And so the program itself is run by Smithsonian, as you probably can tell, but we really rely on our network partners around the globe to help collect data by our side. And so we really rely on them to help us kind of achieve our mission. And so these are the sites on this map. The red sites are our network partners. The blue sites are different Smithsonian sites, and they're actively collecting annual data alongside of us. And so we do two main things, two broad things. And the first thing we do is standardized annual monitoring in coastal habitats. And so that basically tells us what's changing. And so in Florida, in the Indian River Lagoon, which is where I work, we work on seagrasses, oyster reefs, mangroves, and fouling communities, which I'll talk about in a bit. But those listed in gray, the coral reef, soft sediments, kelp forests, et cetera, those are other habitats that all of our network partners work at, depending on where they are in the world. And so we try to work in as many habitats as we can, because there's a lot of habitats in the ocean, and we want to try to work in all of them. Now, the other thing that we do is we do these regional or these global experiments, and these are really important, and these are probably the most interesting things that nobody is doing things like this. And we utilize our network partners, and we do these really simple kind of global experiments that gets at why things are changing. And so the monitoring tells us, well, what's changing? Our seagrasses are some of these things increasing or decreasing in their abundance, and that's what the monitoring tells us. But the experiments kind of tell us why things are changing, and so it's really important for us to do these experiments. We do them every year. We're always doing something different, but we're doing them on a really large scale, and that way we can sort of make generalizations on a global scale on how things are changing, and that's really exciting. And it's one thing that nobody else in the world is doing, and it really makes our program sort of stand out. So as you can tell, our focus is on biodiversity, and so to me, biodiversity just means the number of unique things in a particular area, and the definition can change depending on who's doing the research. And so, for example, biodiversity can be differences in DNA. So we think of genetic biodiversity or genetic diversity, which turns out is incredibly important in the ocean. Diversity can also be functional traits. And so a functional trait is basically how a plant, animal, or algae can make a living. So you can be a predator, an herbivore, you can have a certain morphology, and so that can be a functional trait. So we can think of diversity in that way. Lastly, probably the most familiar for people, is diversity is basically the number of species in a given area. And so that's what we're all probably most familiar with. So I'm going to use this house as an example of our ecosystem. And I use a house as an example all the time because I think it's relatable to most of us, and it's a good example of what can happen to our ecosystem if we don't take care of it. And so think of all the nails in your house as different species. And so there's large nails, there's small nails, but they all contribute to keeping your house a safe place to live. And so this is essentially our healthy ecosystem. And so when we think of a healthy ecosystem, or a nice shiny house like this, we want it to have very little human impacts. And as I've already mentioned, that's hard to find these days, no matter where you are in the world. We also want to have a lot of biodiversity, and that's relative to where you are in the world. So that in the tropics, there's going to be more of biodiversity, which is fine. And in temperate regions, there's going to be less biodiversity. So it's dependent on kind of where you are. But we also want to have a healthy population size of those different animals. And so we want it to be running efficiently. Now, biodiversity is what we study, and so I'm going to stick with that. And so biodiversity, it's important because it supports a lot of different things in our ecosystem. And probably one of the most important things that it supports is resiliency. And so when we think of resiliency, we think of little isolated events that could affect our house or our ecosystem. And we want our house or our ecosystem to bounce back from those. And so if there's a storm, we want our system to bounce back from that storm. We want our house to be fine after the storm's over. So that's something that biodiversity supports, and we've shown that over the past couple of I think decades now. Research has been coming out to look at ecosystem resiliency in terms of biodiversity. Our biodiversity also supports ecosystem functions, and so when I think of ecosystem functions, I think of a really sort of a harmonious recycling program. So there's a lot of nutrient recycling going on. There's another ecosystem function is our primary production. So phytoplankton are the base of the food chain. Another ecosystem function is something like decomposition, which is incredibly important. We need to break down a lot of those nutrients to make them available for other things. Probably the most important for humans, biodiversity supports what we call ecosystem services. And these are going to be things that an ecosystem provides to humans. So that's going to be food, it's going to be clean water, it's going to be oxygen we breathe, it's going to be wood for building our house, and it's also going to be for carbon storage, and there's a variety of things that biodiversity provides to humans. And so we want to in turn support that. So what affects biodiversity? Well, we call these drivers, and the important point about these is that these drivers, they're going to be persistent. And so something like changes in temperature, if it's persistent, it's going to start to pull nails out of our house. So we're going to start to lose species because their distribution or their reproduction is going to be affected by small changes in temperature. Habitat loss is sort of a given, right? If you lose your habitat, you're going to lose species in that. Eutrophication, which is just excess nutrients, that's got a huge effect on the ocean. For instance, phytoplankton blooms are a cause of eutrophication. Also, in the news recently, everybody's talking about this big sargassum blob that's coming towards the Caribbean and Florida. We don't necessarily know what's driving that, but a lot of researchers are suggesting that it's eutrophication. There's excess nutrients that this big algal blob is taking up, and it's making it bigger and bigger than it covers our beaches. Non-native species, that's pretty important. That's a persistent cause of declines in biodiversity. You get these non-native species coming in, and they're incredibly competitive, and they can outcompete a lot of the native species. And so you're losing your nails in your house that way. And I can go on and on. There's a lot of drivers here that are affecting our healthy ecosystem. But if they're persistent, then our ecosystem is going to start to get degraded, and the integrity of our house is going to get degraded, and it could get to the point where we're not going to want to live there anymore. And so prior to this happening, we call our conservation biologists, and we want them to conserve certain areas that we feel are important. And if that doesn't work, then we start to call in our restoration biologists to restore these ecosystems to try to get them back to a functioning state. Now, how do we know that biodiversity is on the decline? Well, there's a lot of papers going back many years talking about how global species are slowly declining. And this top graph is just, these are statistical models going back from the 50s up a little past, looks like 2010, maybe 2020. And they're showing the percent number of species that are declining. And so there's a consistent decline. These models are pretty robust. They're pretty accurate. And then if you look at that little map there of the world, it's showing you where we expect to see declines in species, and those hotter, the warmer colors are where we expect to see more species loss over time, and we're seeing that. And so we have a lot of models like this showing species decline on a global scale and not just locally. So we're pretty confident that we're losing a lot of species, even though they're sort of hard to track. Another point is that we're seeing an incredible rise in the amount of dead zones along our coasts. And I'm sure a lot of you are familiar with the dead zones in the Gulf of Mexico. We've been hearing about that for a while now. These are just areas where the oxygen in the water gets too low where it can sustain life. And so all animals, whether you're terrestrial, or you live in the ocean, you need oxygen. And so as you can see, this graph is showing over time from the early 1900s up to 2010, the amount of dead zones is increasing, and it's sort of, it's all over the world. And so that is causing a lot of species decline, because there are coastal places where a lot of these species just can't live anymore. So moving back to a sort of a lighter topic, where do we find biodiversity in the ocean? Well, it turns out we like to categorize things, and so we call these habitats. And so there's a lot of coastal habitats that the program I work in, there's a lot of coastal habitats that support a high amount of biodiversity. And so it's going to be salt marshes, rocky intertidal coral reefs, there's kelp forests, there's oyster reefs in the middle, mangroves, seagrasses, soft sediment. Mud is an incredible place in the ocean to find a lot of cool animals. But this last picture here on your bottom right, that's got to stand out to everybody, because that's not a habitat, right? That's a dock, and that's made of wood. It's a human constructed thing that's in the water. So we're gonna spend some time on this. And so this is what I'd call an artificial structure. And basically it's any sort of infrastructure that humans create that we put into the water. And so something like a dock, a marina, bridges, etc. And so a few of these here and there, probably not having much of an impact. We probably don't notice them. They're probably good for maybe a good refuge for fish. As you can see in the upper right here, there's a lot of fish hanging out underneath this dock. So they're not all that bad at first glance. However, this can get a bit out of hand. And so we call this shoreline hardening. And this is on a landscape sort of scale. So this is big scale. Excuse me. So the reason why we put up seawalls or concrete or whatever along our coastline is basically to protect coastal communities. And so people want to protect their property. Cities want to add marinas or create a landscape where people can live closer to the water. And so this is a problem because it's there's really not a lot of regulations on what city municipalities can or can't do. There's not a lot of permitting, especially when you're a city and you want to put in a seawall. You don't really have to do any sort of scientific assessment on what's happening. So this is basically replacing a lot of the coastline with artificial structures. And so there was an older paper that came out maybe 10 years ago now, and they had done a global or an analysis of the US, and they came up with 15% of our coastline is hardened. And that's not docks, that's not bridges, that is literally putting up, like that picture in the middle there, a seawall on your shoreline. And 15%, that, to me, that's a lot, especially when you think of the whole US coastline. But when you move into urban areas, cities, more than 50% of the coastline is going to be hardened. And that is a lot. And that can have a lot of negative impacts on the coastal habitats that I went through. Now there's been some studies, some global studies that have looked at how seawalls compare to natural habitat in terms of the animals living there. And one of the studies that I looked at the other day said that seawalls have about 15% less diversity, so the number of species, as compared to an adjacent natural habitat. So think of, if you find 100 species in a natural habitat, 15 of those are you're not going to find on seawalls. And so that's not a huge difference, but it's really where the abundances are that's kind of striking. And so on seawalls, these researchers found that there was 50% less of the count of these species. And so that's a lot. And so you're cutting down your population by half when you put in these seawalls. And now this is very site specific, as you're going to see in a couple of slides. This doesn't happen everywhere. It's seawalls, they have not just seawalls, but artificial structures can have both positive and negative effects on the marine flora and fauna, which I'll show you in a second. So it gets complex when you start to look at individual sites. But to make this, I like to take kind of global observations and see how it pertains locally. And so what I did was I just took a map of Fort Pierce, which is where the Smithsonian lab is located. And that yellow star, that's where I am right now. That's where our lab is. And this is the Indian River Lagoon. And then to the right there is the Atlantic Ocean. And so that's our little inlet bringing oceanic water into the lagoon. And so what I did was I randomly drew a box around the area. And what I did was I went through and I highlighted all the spots that were hardened. And I was pretty generous on what I thought was hardened versus what was natural. And so this is definitely an underestimate of what's hardened around the area. But the coastline around this box is about 50 kilometers, and a kilometer, for those of you who don't know, is a little more than half a mile. Turns out about 20 kilometers of this is hardened. And so that means that about 40% of this coastline is going to be hardened. And that kind of fits with our 50% of an urbanized area that's going to be hardened. And so it turns out that a lot of things that we study, like seagrasses and oyster reefs, well, they don't like to be near these hardened areas. They have changes water flow. It does a lot of different things that affects these particular habitats that hold a lot of the biodiversity that we're interested in. And one of the things that we do, and one of the things that we're actually starting this summer, which I'm excited about, is that we're going to start to map our areas, the sites, the MarineGEO sites that we all work at. And so basically what somebody could do is that you make a series of categories like salt marsh, beach, mangrove, dock, riprap, etc. And then you just highlight them, and then every couple of years you do it again. And then over time you can see changes in the shoreline. And then once you couple that with some of our monitoring data, you start to get a good idea of why some of these hardened shorelines are not very good for the habitats that we work in that support high biodiversity. So this is a good local example. I didn't know before I made this how much of our coastline was affected by humans, but it turns out it's a lot of it. So I wanted to show you a quick little video of, let's see. So this is just a dock piling, and I put a video camera on, and we every once in a while we'll motor around in the boat and we'll film what's growing on these dock pilings. And as you can see, this is there's a lot of different animals on this dock piling, and so there's hundreds of different species on here. And so I wanted to show you this video because sometimes it's hard for people to see a dock and think, oh, there's animals growing on there. Well, this is a random piling, and clearly there's a lot of different things growing on here. So it makes for a really interesting habitat to study, at least from my perspective. And so it turns out that it's relatively easy to study, and I've been studying these types of habitats for a really long time now. And so I wrote a protocol on how to study artificial habitats on a global scale, and I've wrote several protocols on how to do this for different habitats for MarineGEO. And this one was perfect for me because I really enjoy working in this type of habitat. And so the way that we do it, it's incredibly simple. You don't need to get into the water. All you really need is these small pieces of plastic, and this is just a PVC piece of plastic. We call it a fouling panel because fouling communities, which I'll show pictures of in a second, grow on these things. And so it's 13 by 13 centimeters, so it can fit in your hand. We attach it to a brick to hang it from a dock so that it stays stable. We can attach it to PVC, and we can put it in any environment or any habitat in the ocean. And what's really interesting about marine animals is that if you put something hard in the ocean, you're going to get stuff growing on them. And so it happens really quick too. A lot of these animals have really short lifespans. And so you get a lot of stuff really quickly. And these are some examples of what we call fouling communities on these panels. And they're quite colorful. There's a lot of different species on here. A lot of stuff that most of you probably have never heard of before, but they make up 99% of the animals in the ocean. And it turns out there are predictable patterns on how these animals develop on these pieces of plastic, and we call that succession, and it's a common ecological theme that we like to study. And so it's basically how these communities develop over time. And so we've been working in the lagoon now long enough that we know the story. We know what comes in first. We know who dominates these communities. We really have a good idea of how they get assembled over time. And so that's advantageous because that can give us it gives us a baseline of how we can start to study these communities and how we can use them to look at sort of different ecological themes, which I'll talk about in a second. So what I wanted to do is I wanted to spend the last couple of slides talking about some applied examples on how we study fouling communities, how it relates to biodiversity and human impacts, and specifically artificial structures. And so I'm going to give 4 different examples of stuff that I've done in the past, which each one of these touches on a different theme in ecology, but taken together, it kind of gives you a nice holistic view of some of my research and some of the different ways that we can use these communities to ask very different questions. So the first one is long-term monitoring, and that's pretty common in science. We like to study things over a long time period. And frankly, I think some of the best questions come out of these long-term monitoring studies. You get to know your system really well, you get to see changes, and then you can ask really interesting questions on why those things are changing. And so it's a really good way to get a baseline amount of data that you can start to use to manipulate and look deeper into. Then the next one is going to be a comparative study. So that's relatively simple. And what we did was we just compared artificial habitats to natural habitats in terms of biodiversity. The next one is fundamental ecology. And so this is my favorite. I love doing predation studies. And so I love to see who eats who and how that affects what the community looks like. And the last one is it's a clever idea that we came up with that we did looking at a different type of ecosystem function. And it was a community feeding study, which I'll show you, which the results came out really, really nice. So the first is long-term monitoring, and we've been doing this since 2017, which is about the same time as when MarineGEO started to collect data at all of our different sites. Here we have 6 different sites locally that we go to every summer, and we put out 6 of those panels at each one of these sites. We have 3 docks and 3 seagrass sites, and this is kind of a comparative study because we're looking at 2 different habitat types, but the idea is that we're doing sort of a long-term monitoring. And so we wanted to have a human impacted site and we wanted to have a natural site. And so that's why we used seagrass beds. We do this every summer from June to September. So on June 1st, we go out and we deploy all of our panels. And then every 30 days, we'll just take photos of these panels. And that's the major way that we collect data. And after 90 days, which is our full summer, we'll bring everything back in. I'll put it under a microscope. I'll ID everything, and then do some other stuff with the critters once they're in the lab. The outcomes with it, it's pretty simple. We take these photos and basically we just estimate % cover of the species living on those photos or living on those communities. And so we can look at, who's dominant in the different habitats, and we can look at that, over time. What I'm really interested in is long-term changes in these communities with changes in the environment. So we know that temperature is increasing over time. We also know that these animals are tied really tightly to temperature. And so if temperature is increasing over time, we should see changes in our communities over time. And even though we started in 2017, we still probably don't have enough data yet to look at that. We haven't had enough differences in our summer cycle. But we expect things like if it's a rainy summer, we should see differences. If it's a dry summer, we should see differences. If pH over a long period of time is changing, we should see differences in that. And so it's a really nice monitoring tool that we can look at local changes in these communities, but we also can think kind of on a larger scale, looking at big changes in our water and not just just thinking in terms of large changes. And a lot of different MarineGEO sites are doing this exact protocol. So we don't just have stuff locally, we have stuff from a lot of different sites around the Americas, at least. One incredibly useful thing that we also get out of this is we're monitoring for non-native species. And it turns out that non-native species love to live in artificial structures. And there's a variety of reasons for that. Every year, new experiments are being done to look at why that's the case, but it's a global phenomenon. And so we see it from the poles all the way down to the equator, that non-native species, they really like to live on artificial structures. And so monitoring these communities at docks, we really get to see who's coming in. And then after working here for so long, we get to know, we'll automatically know if they're not from around here. And I can tell you, every year I see something different that I have never seen before. And so it just, things are moving around a lot, and this is a great tool to look for non-natives. And so we're actively doing that as well. So now moving to more of an experimental approach, we wanted to do a comparative study on how artificial structures are different from a natural habitat. And we live in Florida, and there's a lot of mangroves in Florida. And so for those of you who don't know much about mangroves, they live along the shoreline. They have these really cool, what we call prop roots. And you can see that in the picture. And these prop roots come off the tree and they go into the water and they go down and they bury themselves in the sediment. And so turns out there's a lot of cool animals that like to live on these roots. And I'm sure a lot of you have seen pictures of these really beautiful communities growing on these roots. And so it made sense to me to compare the communities on these roots with those of a dock piling, which is also wood. So it's one of the few instances where we can make sort of an analogous comparison between two different types of habitat that are relatively similar, one being artificial and one being natural. And so you can see on the map there on the left, these are all the sites that we visited over the course of 2 years. And so every 3 months, we'd go back to these sites and we'd collect our panels and we'd collect our data, bring it back to the lab. And so what I'm going to show you is data on species richness. And for the next couple of experiments, we're just going to stick with species richness because it's really easy to understand. And it's the number of species living in your community. And so it's your diversity, essentially. So in this first graph here, the black circles, those are our artificial habitat. The white circles are our mangrove habitat. And this is combined for all of these different sites. And so besides that one little spot in January of 2016, we're constantly seeing higher diversity in artificial habitats. And that kind of goes against what a lot of the previous research has suggested, that, well, artificial structures, they shouldn't have a lot of diversity. Well, we're finding a lot of diversity in our structures, and there's a reason for that, which I can get into at the end if there's time. So constantly over the course of 2 years, besides that one little point there, we saw more diversity in our artificial habitats. Now, as I mentioned, non-natives really like to live in artificial habitats too. And so this is a graph on your right, the same time period, and it's besides that one little point, again, it's showing that each time we went out, the trend is that we're finding more non-natives in our artificial habitats. And one of the things we did was we subtracted the number of non-natives from our natives, and we looked at just native species richness, and it doesn't change. Overall, species richness is always going to be higher, regardless if you have more non-natives. It's always going to be higher in your artificial structures. And so when you add the non-natives, it doesn't make the two habitats similar. So take home for this is that in Florida, we're seeing a lot of diversity in our artificial structures. So moving on to one of my favorite experiments, this is we call it a biotic interaction. And so this is what we call a caging study. So it's basically looking at predation. And it turns out, and one of the reasons why I love doing these types of studies is that predators, let's say consumers, so predators or herbivores, they have an incredibly strong impact on communities. And so they shape the way that a community looks. And so when you go out and you're looking at whether it's your garden, whether you're walking through the forest, herbivores and predators, they have a really strong impact on what you're actually seeing. And so we like to test that. And so one easy way to do that is to take one of our panels and we put it in a cage. And what that does is it prevents, in this case, fish, which are our main predators. It prevents fish from consuming or eating the stuff in our communities. And so here's an example from the study. And on the left is a panel that was in a cage, and it's got a lot of this. These are ascidians, and they're solitary ascidians, and so they're incredibly common in the ocean, but they're soft. They're really competitive, so they're weedy, but fish like to eat them. And so when you put a panel like this in a cage, you're always going to get these ascidians growing on them. And when you don't have a cage, you typically get barnacles, and that makes sense because a barnacle is, it's really hard. Not a lot of fish want to eat a barnacle. There are a couple in Florida that do, like puffer fish, There's sheep's head. There's a couple of different species that do like to consume barnacles, but in general, you're going to get communities that are going to be more, they're more calcified. They're hardened communities because our fish don't like to eat them. And so we did this in seagrass habitats, and we did it in artificial habitats. And so we wanted to compare predation in the two different habitat types. And so again, we're just going to focus on species richness because that's relatively simple to understand. And so we wanted to look at predator effects on these communities, and we did it in the different habitats, but we also did it with different ages of communities. And so we did it with communities that were 30, 60, and 90 days old, because we thought that, maybe an older community with larger prey would be less susceptible to fish. And so you can reach a size refuge where a fish, they're just too big, so a fish wouldn't want to eat you. So we like to use age of community as a factor in when we're looking at some of these, doing some of these experiments. And so what this graph shows you, and so you have species richness, and so we have 0 species up to 20 species in these communities. Now the dark bars are going to be our artificial habitats. The lighter bars are our seagrass habitats. And the first set of 3 are in cages, and the second set of 3 are out of cages. And so these are, so you can see cage 30 days, cage 60 days, etc. And so what we found was that, of course, there's going to be a lot more species in artificial habitats as compared to seagrass habitats. That's incredibly clear here. We also found that there's a lot more species when they're in a cage versus out of a cage, which makes sense because the fish are consuming species, and you expect to see a decline. In seagrass habitat, we don't really see that signal though. So there's some indication that there's more species in these caged communities, but overall they look relatively the same. And so there's something going on here, and it's interesting. And so we've been exploring that a little bit further, but overall, more species in artificial habitats as compared to seagrass. And so we saw that with the previous slide. And then there's more species when predators are excluded. And so, so that's interesting. And so we're starting to develop this story here about how these communities are working. And now we're integrating how predators are affecting the way that they look. And so, as I had mentioned, in terms of native and non-native species, we wanted to look at how predators affect those. And so introduced here is just another word for non-native. It just means a species that's been introduced to a new area. So sometimes I like to call them introduced, sometimes I call them non-native. They're interchangeable. So here again, we're looking at species richness, and so 0 to 8 different species. And it turns out more non-natives are in artificial structures, which we expected because from what we've learned in the past experiment. But what's interesting is that when we brain fart, predators are reducing the amount of non-natives. And so this is really important, and we call it biotic resistance. And so what that means is basically that when you have a good diversity of predators, they're going to eat all these non-native species, and they're going to keep them in check, which is really important, because one of the things that we worry about with non-native species is that once they get into an area, they can spread and they can outcompete all the native species and just take over the area. Well, what we're seeing here is that we're seeing predators consume a lot of these non-natives, which is really important. We don't know which ones they consumed. We just, but they're consuming some of them. And so the last experiment that we did, so this is an ecosystem function experiment, and it's a feeding experiment. And it's a pretty clever way that we came up with on how we could look at how diversity in these communities relates to ecosystem function. And so what we did was we took our panels and we put them in the Indian River Lagoon in particular places where we knew we'd get a few species or we knew we'd get a lot of species. And so what we're doing is that we're creating these communities that are either really diverse or really not diverse. And so we left them out for, I forget how long, but several months, so that even if a panel had 1 or 2 species on it, the panel was still covered with that species. So the same amount of biomass is in these different communities. And so what we did was we went out and we collected all of the communities, all the panels, and there was 30 of them. And we brought them back to the lab, and then I put them under a microscope and I counted all the different species. And it turns out that we had anywhere from 1 to 30 different species on these communities. And these are filter feeding species. These are species that would have an effect on water quality. And so in the lab, we put them into little separate containers, and we fed them different phytoplankton species. And we fed them a large one and a small one to look at the differences in the size of the prey. And we measured 2 different things. And the first thing we measured was clearance rate. And this is the graph on the left. And clearance rate is basically just how much water is being filtered out per minute. And so it's a really nice way to look at ecosystem function. And so we're using our species richness on the bottom, so 0 species. So we used panels that had nothing on them, all the way up to 30 species, and this is our clearance rate. And as you increase your diversity, you get this really positive line. And so the more species you have in your community, the more they're filtering the water. And that's really important because that goes against, well, for instance, artificial structures in Florida, they have a lot of species on them. And so this is a positive side to having artificial structures because there's a lot of species on them and they're filtering out the water better than communities that don't have a lot of species on them. And so it's a really nice result. It came out really nice and we're really excited about it. This graph on the right, this is what we call retention efficiency. And this is basically just the percent of the food that was lost over time. And so 100% would be all the food, all the plankton were eaten. And then, the lower amounts would be less eaten. And so as we increase our species richness, more food is being eaten over time. And so that makes sense because, clearance rate is, they're both correlated. They're both very similar. And so these are really exciting results because it shows that diversity is really, really important to ecosystem function. And so the more species you have, the more water is going to be filtered out. And so I think, we really need to stop focusing on individual species, like for instance, oysters or clams. We need to start thinking about larger, we need to think about how biodiversity as a whole can affect our water clarity, because a lot of these species, they're better at filtering out the water column. And when you put a community together, they're doing an incredible job. And so they're helping, I guess, reduce the eutrophication or the pollution in our estuaries. And so this is a positive spin, and I wanted to sort of end with this experiment because it's a positive spin on artificial structures. And so to wrap up, a couple of take-home messages, and I'm sorry about all the words. I tried to have pictures on each slide for people to look at, but these are sort of the take-home messages. And so this is kind of the important stuff. And so to end with, our coastline, and this is a global thing, is heavily impacted by infrastructure. And we don't know around the whole world how much of our shorelines are hardened, but a lot of it is. And it's a problem because it's replacing natural habitats. And these habitats are important because they hold a lot of our biodiversity. Now, people are still, reports are still coming out that people keep moving towards the coastline. They're not moving away from the coastline. And so we're just going to continue to get increases in shoreline hardening. And there's really not a lot of restriction on where people can put those in. Now, also, I just focused on biodiversity because that's what I work on. But there's a lot of other things going on in terms of artificial structures. It changes a lot of the physical qualities of the water. It changes water currents, it changes depth, it changes a variety of things. And so there's a lot of other things going on here that I skipped over. But I just wanted to make the point that it's not just about biodiversity. So globally, artificial structures, they vary in their impacts on marine species, and we're still learning why these variations happen. In the temperate regions, it's very different from the tropical regions on the effects of these artificial structures have on the communities that we're looking at. And so thankfully, with my program, that's something that we can start to look at, and we can start to answer those questions, which I'm really excited about. Now, in the Indian River Lagoon, we see increased diversity on our artificial structures, but we also see more non-natives, and so it's sort of a double-edged sword there. We like having more diversity, but we don't want the non-natives, and so I don't know how to do one, to get one but not the other, and so that's still a challenge. And globally, we're finding non-natives on artificial structures, and for the most part, these non-natives are restricted to these artificial structures. They're not living on a pine, and they're not moving to a mangrove. They're not moving to a seagrass blade. They're not moving to a rocky intertidal area. They're kind of stuck on these structures, these human-built structures, which is a good thing. Now, there has been, in the past 10 or 15 years, there's been some green ideas, green initiatives, and we call these eco-engineering, and they're basically modified infrastructure that we can put in the water to support biodiversity. So instead of putting a flat seawall in, you can put something that's got some crevices and some nooks and crannies that will support increased biodiversity. So at least you're doing something to help the marine animals. So we want to, as I mentioned in one of the first slides, we want to get those abundances back up to where they were, because of all those reasons why biodiversity is important. The last two, I just wanted to highlight using fouling communities as a really good tool for as our experimental unit to look at kind of basic themes in ecology. We can use these communities to do a variety of different textbook kind of examples of how the ocean works, and they're incredibly useful. They're not necessarily what people would think of when they're doing when you're learning about how the ocean works, but they're really useful, and it's been proven time and time again. And lastly, using these communities is really important to our monitoring. They tell us a lot of things about how our system is working, and so it's a great way to look at changes in the environment, especially long-term changes like temperature rise. It's going to have a significant effect on the species that we're seeing, and if we're collecting data year after year in a simple way, just taking photographs, we're going to be able to see those changes, and then we can correlate them and then have a really good idea on how temperature, for example, can affect a different marine species. And lastly, just using these to study the non-natives is incredibly useful. It really gives us a good idea on how those non-natives, how competitive they are, how they have the ability to outcompete some of our native species. And so it gives us a really good way to study their natural history and learn a little bit about them, and to find out when they come into our area. And so thank you for listening to me for an hour. I wanted to leave you with some pictures of some of these critters that live on our panels. And so see, these are some of the close-up things of some of these really incredibly cool animals. They do a lot of different things. There's a lot of different types of them. So you'll see more animals on one of these panels than you would going to a zoo or an aquarium, and they make their living in incredible ways, and they've been a real pleasure to study. So with that, if there are any questions in the chat, I can take those or try to answer those.
There's two, Dean, that I left for you to answer from Lisa Mulcahy. The first was, are high levels of ascidians indicative of a lack of herbivores in natural environments?
Well, so it'd be a predators, right? And so not herbivores, but that's one of the things that we're interested in. So one of the reasons why we do these caging experiments is that we want to get a baseline of what predators are there. And so, if we continue to repeat these experiments over time, we should see that the things that like to get eaten are going to be there regardless if we put a cage on or not. And so I think it depends. There's these species can be found, they're not found everywhere. They're found in a lot of places, especially in estuarine environments. They range from these ascidians. They range from the poles all the way down to the tropics, and so it's a good question, and it we need that baseline data. So it would be easy to test if you put a cage around these things and you don't let anything eat them, would they start to show up? And so or there's a lot of ascidians that also have chemical defenses, and so some of those just don't like to be eaten because they're really tough and fish don't like to eat them, or they have chemicals inside of them that prevent fish from eating them. And so it's easy to figure out just by doing some simple experiments. And so but each site is different, and so it's not a pure correlation that one would be there while the other isn't.
And she has one other question. I'm not sure if you can answer this, but why are there so many filter feeders in mangrove environments?
Well, so the mangroves provide substrate, so their roots provide substrate for these animals to live on. And so a lot of these animals that we study that live on these panels or on mangrove roots or on pilings, they're called sessile. So that means that they attach permanently like a barnacle, and they filter feed. That's how they make a living. And so what mangroves are doing is they're providing a lot of substrate for filter feeders to live on. And so that's why you see a lot of sponges or bryozoans or ascidians on mangrove roots, is because the mangroves are providing substrate for these things to live on. And so it goes back to habitats and why we take a habitat approach to studying these things, because habitats like oyster reefs, seagrasses, coral reefs is a great example. They provide substrate and refuge for these things to live on. And so the mangrove roots are providing substrate for these filter feeders to live on. And filter feeders are found everywhere in the mud. You're going to find lots and lots of filter feeders there. It's a common feeding mechanism in the ocean, and so but they're there because more or less because of the substrate.
We've got another one that just came in, and since we're hitting 10 o'clock, we'll make this the last question. But Samuel Shames is asking, considering that artificial structures are beneficial to non-native species, is it a good idea to create artificial reefs?
So artificial reefs, people put artificial reefs in because they do it for fish. I haven't seen any good data that suggests that fish are on the decline, certain fish that they're putting these reefs in. What artificial reefs are doing, and there's plenty of papers out there that show this, is that they're providing habitat for non-natives. And so the same thing goes with, we see this a lot in Florida. There's a lot of people, not a lot of people, there's people who take these really large ships and they sink them off the coast. And they claim it's because they're providing habitat for fish and for things. I think it's an awful idea. I don't think you should put anything in the ocean that big. There's no good reason for it. There's no strong data to suggest otherwise. I could be wrong, but I think that artificial reefs are providing more habitat for non-natives than they are benefit for fish. There's plenty of places out in the ocean that fish can congregate that are natural. And a lot of these artificial reefs are actually supporting a lot of lionfish around the area. And so You can swim out, you can snorkel out to some of these artificial reefs, and you're going to see a lot of lionfish. They're certainly they're non-native, they're invasive, right? They have a negative impact on communities. And so once you have a negative impact on a community, we start to call them invasive. And so lionfish are notoriously invasive. They eat everything. And so artificial reefs providing more food for lionfish, well, they sure are. But a lot of the things that I study living on these artificial reefs, they're non-native, and there's plenty of papers out there that say that. So in my opinion, no, I don't think that artificial reefs, I think there's more effort. More effort can be put elsewhere rather than putting these things out. But that's just my 2 cents on that.
All right, well, I think that will wrap us up for today and for this season. Next year, we'll do this all again. So thanks, Dean, for talking to us today about your work.
Thanks, everybody, for listening.
Yep. And with that, we'll sign off. Thanks, everybody.